And let's see if I can get this to work. Please work. I see they fixed the teaching station here. Well, that's interesting. Oh, there it is. Okay. I'm going to stop share because that's not the one I want. So those of you who are zooming in, are you seeing one big screen, one big slide, or are you seeing my notes view?
We see the notes.
All right. How about now? Do you see the one big slide?
Yeah.
All right. That's what I would prefer. It's easier for you to see it online. Okay. So before the long weekend, we left off thinking about the physiological processes of microbes. And this is kind of a real big picture idea of several concepts all onto one slide. And so thinking about the idea of glycolysis, which we can do, and microbes can certainly do, many microbes. All right, so glycolysis is where you typically think about running glucose as the starting substrate through a series of reactions. And the end goal is to get a little bit of ATP. And then I'm going to talk a little bit more about this NADH idea. And then the other key thing about glycolysis is you come down to the point in stage 2 of glycolysis where pyruvate is made. And that is what I like to think of as a decision point by the microbes. And it really all depends on whether or not there's oxygen available in the environment and whether that microbe can make use of oxygen, what they're going to do with that pyruvate. So let's imagine that there is oxygen in the habitat and the microbe like E. coli can use oxygen. So if you're a smart E. coli, what you'll do is you'll take the pyruvate and shunt it over to the TCA cycle. So here, this middle part is, I kind of consider all of this the TCA cycle. They broke it apart a little bit here where they're showing pyruvate getting converted to acetyl-CoA. That's the very first step leading into the TCA cycle. So when you push pyruvate into the TCA cycle, you get, again, these NADH molecules, these electron carriers. I'll talk about them. And then you see the cycle, the TCA cycle or the Krebs cycle. And you can see that you get the breakdown of glucose. So if we start with glucose, what they like to say is if you follow this pathway through the end of the TCA cycle, you get the complete breakdown of glucose to CO2, which means you can't really break it down any further than that. Glucose is completely broken down. You got the most bang for your buck, so to speak, in terms of being able to completely break down glucose and get some necessary ATP and then these electron carriers. Then you see on the far right of the diagram this electron transport system or electron transport chain. And they have the equation here with oxygen. So they're saying oxygen is going to be used in this process now of the electron transport chain. Oxygen is what we call the final electron acceptor and will essentially, they're not showing details, but you have electrons being passed down the chain from donor to acceptor, donor to acceptor. It's kind of like hot potato. They just keep passing the electrons down until they reach oxygen. Once oxygen gets enough electrons and protons, it will form water. But that's not the end of the story. Whenever you think about an electron transport chain system, you have to think about really two parts to it. Yes, you have electrons being transferred from donor to acceptor on down the line. But in that process, you have protons building up across the membrane. All right, so there, I wish again that the artist had put some more protons here, because there should be lots and lots of protons building up outside the cell so that you get a proton gradient established across the membrane. All right, so I can't really see it, but those lots and lots of protons. And so now as those electrons are getting passed from donor to acceptor, there's a little bit of free energy at key steps in that pathway that allows for pumping of protons across the membrane to the outside. So on the right here is the outside of the cell. So electrons get passed, protons get pumped across the membrane. Electrons get passed, protons get passed across the membrane. So it's a concept that goes together. You can't divorce the two. When you have that electron transport chain system, you'll get protons being pumped across the membrane. So then you get this proton gradient, which is great because that's a source of potential energy or free energy available to do something. And depending on the microbe and their needs, they can use that proton gradient for a variety of things. But one of the key things you're seeing here is that you have the protons starting to diffuse back across the membrane by the laws of diffusion. The protons want to diffuse across to even out across the membrane, but they have to go through the ATP synthase to do that. And as they do that, it's a little molecular motor, and actually the flow of protons through there actually causes a rotational torque force on these subunits that allows for ADP and inorganic phosphate to react, and you get ATP. Lots and lots of ATP through the electron transport chain process. All right, so oxidative phosphorylation is the term that we give for this idea of an electron transport chain tied to a proton gradient and lots of ATP being produced. So phosphorylation is referring to the idea that ADP gets phosphorylated to make ATP. Now, oxidative, when you hear the word oxidative, does it make you think of, what does it make you think of just in general? Oxidative. Oxygen, right? That's, and I can totally get that because my brain goes there first too. However, in a microbial world, I need to caution you, oxidative does not mean oxygen always in the microbial world. You have to look at that word oxidative or oxidation in its strict definition, which just means stripping of electrons off of a molecule. That's what oxidation is. You grab electrons off of a molecule and separate the electrons off of it. That's oxidation. Reduction, on the other hand, is putting an electron onto a molecule. So when we say oxidative phosphorylation in the microbial world, could it mean that oxygen's involved in the process? Yes, and that's what we're seeing here. Oxygen is there. However, there's plenty of microbes that do oxidative phosphorylation, and oxygen is not involved in the process. So you have to kind of be nitpicky about that word oxidative in the microbial world. It does not always mean oxygen. And I'm making a big point about this because last time I taught this class and I asked questions about the physiology portion, everyone kept saying oxidative meant, oh yeah, there's oxygen involved. Well, not exactly. All right, so be careful about that. We're used to thinking that way because we use oxygen. We're humans, we use oxygen, and so we kind of get that stuck in our head. But be careful about it. All right. So we talked about ATP last time, how the flow of protons through there generates that torque force, that rotational force. And we got into this idea of fermentation, how glycolysis is an example of fermentation, where if you have E. coli, for example, that is in an environment without any oxygen, it will go ahead and one option would be to run fermentation. Because you do still get ATP produced through substrate level phosphorylation. It's just not, you don't get a whole lot, but you get some. And so it's an option for a lot of microbes. And if you don't have the option of oxygen being around you, or you might be one of those microbes that just doesn't use oxygen, then fermentation is definitely a way to go to keep alive and at least eke out some ATP and keep going. So substrate level phosphorylation, we talked about that last week, how it's the substrates involved in this step-by-step pathway that is going to donate off a phosphate to ADP. So it's substrate level. It's, in this case, substrate C is donating off the phosphate to ADP. So it phosphorylated ADP to make ATP. So glycolysis is an excellent example of substrate level phosphorylation. So if you're a microbe without oxygen, this could be a way to keep alive. So one thing on here, so we talked about this last week, how there's some nice ATP being produced during stage 2. One thing about stage 3 that I want to kind of hone in on and make sure you're aware. So here you see a couple of pathways where they show NADH going to NAD+. Then up here you see NADH in stage 2, it says going to stage 3. So what they're trying to show here is that when you're a microbe, or pretty much any living cell, there's not a lot of NAD or NADH in a cell. So it has to get constantly recycled so that you have some NADH, but you also have some NAD+, because there's not a ton of it in the cell. So there's this need to keep it getting recycled, so to speak. And so here, let's go down to pyruvate, and you see the pathways here of stage 3 of fermentation. Like, we can do this. We can do pyruvate to lactic acid when you go anaerobic in your muscle tissue. If you do bursts of speed and you run out of oxygen and you start to go anaerobic, you can do that, and then you get lactic acid buildup in your muscles. So we do that. So in that process, you have the conversion of NADH to NAD+ to push that pathway to keep it going to lactic acid. Then you see, okay, that now the NAD+ is going to stage 2. So then we go back up to here in stage 2, and you see NAD+ is being used in this pathway, this part 2, this part of the part 2 pathway, stage 2. All right, and so NAD is getting converted to NADH here. And then it says, okay, go back down to stage 3. All right, so they're trying to show that it's kind of toggling back and forth. You use NADH to push this pathway, you get NAD+, which can feed back up to here, allow for this reaction to happen, and it keeps going. All right, so one advantage to doing fermentation for the microbe is to make sure that there is NAD+ getting turnover, so to speak. You have turnover so that you have enough NAD+ to keep this all cooking. All right, so that when they examine what's going on in stage 3 of fermentation, that seems to be one of the common themes, is that the NADH will get used to make NAD+, which can then float back and help with stage 2 reactions. Then here you're seeing just an example of several different pathways, fermentation pathways, and this totally depends on the microbe, really depends on their habitat. What do they have available for them to use in terms of substrates? So you can see that depending on the microbe, they can ferment pretty much anything. They can ferment amino acids, pyrimidines, purines, okay, sugars, of course. So the one we talked about is kind of right here in the middle, down to pyruvate. But here they're showing pretty much any pathway you look at, the goal is to get a little bit of ATP and to recycle your NAD. So you keep that going, all right? They don't really show the NAD on here, but they're at least showing the ATP. So fermentation, certainly we've taken advantage of it as humans. It's a billion-dollar industry, several billion-dollar if you add it all together. If you think about cheese production in Wisconsin, butter production, wine and other alcohols, certainly beer, of course, but also industrial solvents and or organic chemicals that we use are also part of the industry. And so it truly is a multi-billion-dollar industry by us understanding fermentation and capitalizing on it to have the microbes do the work for us. So we don't have to synthetically do this. We just understand how you work with the microbe, give them the necessary conditions to force them into a fermentation situation, and then they'll be happy as clams making all these products for you. And so it certainly it is directly impacting our lives in many, many ways. Here again, just some equations showing the overall kind of big picture idea of some of these different fermentation pathways. And again, some of the key things to look at here is when you look at the final product. So let's look at the alcoholic fermentation. You start with sugars and you get ethanol and CO2. Ethanol is not, so it's not all broken down to CO2 here. So fermentation is not terribly efficient in that regard. To be most efficient, you'd want to take that hexose and totally break it down to CO2. Then you get the most bang for your buck. Here you can see that all the products here, yes, there's some CO2, but there's other carbon-based molecules that aren't all the way broken down. So it's not terribly efficient. That's why you only get a little bit of ATP, but at least you stay alive if you're doing this. And you can see a bunch of different bacteria here on the far column that are capable of doing photosynthesis, fermentation. So it just depends on the microbe and what habitat they're in. Okay. All right. So now let's keep thinking about no oxygen. So a microbe is confronted with no oxygen in the environment, and they could do fermentation, absolutely, if they have that capability. Another option would be anaerobic metabolism. Some people call it anaerobic respiration. That's okay to say it that way too, but I find sometimes it confuses students. So I'm just going to say anaerobic metabolism and try to stick with that predominantly. So what does that mean? And again, AN in the front of a word is without. So without, aerobic means oxygen. So again, we're saying no oxygen involved. So that means that what we're going to see is we're going to talk about an electron transport chain process, but instead of oxygen sitting at the end of the electron transport chain, it's going to be something else. It won't be oxygen. So it's anaerobic. It's typical, well, it really does depend on the microbe, I guess. Many microbes will grab some kind of inorganic molecule and have it at the end of their electron transport chain to be the final electron acceptor. But you can have organic acids serving that role as well. It just really does depend. Microbes are so versatile. There's a whole list of different things besides oxygen that can be used. So it's an electron transport chain process, ETC. So you embed it in the cell membrane of the microbe, and you have that same idea where they're passing electrons down a chain, donor to acceptor, on down the line until you reach the terminal electron acceptor, and it just won't be oxygen. It's going to be something else. If you study these pathways and look at them and look at the thermodynamics of it and say, okay, how much energy, how much ATP can get made from this? If it's anaerobic, it's not going to be as good as oxygen if you have oxygen sitting at the end of the electron transport chain. And that's, we talk about it in general microbiology when we looked at that redox tower. And in the redox tower, for those of you who've had Gen Micro, remember that the oxygen was at the very bottom of the tower. And that is pretty much saying that oxygen is the best electron acceptor that we know of amongst living organisms. When they use an electron acceptor, oxygen is the best. You get the most ATP production if you can use oxygen and use it as a final or terminal electron acceptor in this electron transport chain process. Any other molecule will not be as good. So the microbes will do their modified electron transport chain process, something other than oxygen sitting at the end. They still get a proton gradient though. They link it to a proton gradient. As the electrons are passing down the chain, there's enough free energy to pump protons across the membrane. So you still have that going on. And then the protons will diffuse back across the through the ATPase across the membrane, and you'll get your ATP being produced. Whatever products get made during this process, if it's, depends on what the final electron acceptor is, they don't really care about that. They're just using it to play the game, so to speak, of getting that electron transfer process to kick into gear. They'll just excrete out what's made at the very end. They don't really need that. Their goal is to make ATP during this process. So let's compare. some of you who've had Gen Micro, that this should look familiar. This is a nice comparison trying to get across the point of what happens if oxygen is being used versus something else besides oxygen. So here on the left, you see oxygen here as the final electron acceptor. So the electrons are coming here, NADH is donating some electrons, and some here are the donors and acceptors, all right? And so they're just passing the electrons. You see that in that process, protons are getting pumped across the membrane, and then you get this buildup of protons. And then as they don't show the ATPase, you have to imagine the ATPase here. All right, so those protons will flow through, again, the ATPase and get you your ATP. All right, so this is one example of aerobic metabolism, with oxygen sitting at the end. Over here, however, it's not oxygen sitting at the end of the electron transport chain. In this case, it's nitrate. NO3 minus is nitrate. And that's very common. Some nitrogen-containing molecule is a very common strategy amongst microbes to be the terminal or final electron acceptor. But again, it's the same basic idea, right? If you compare what's happening on the left and the right, you have a lot of similarities. It's still this electron transfer process, which is linked to proton pumping. And then you have to imagine those protons diffusing through the ATPase to allow for ATP production. This NO2 minus, nitrite, is NO2 minus, nitrite. That just gets excreted, just like water would get excreted, all right? So they're not, the goal here isn't to make nitrite. The microbe really doesn't care about that. They're just playing the game, energetically speaking. They're playing the game so that they can create a situation where they get a proton gradient established across the membrane so that those protons will diffuse through the ATPase and get you some ATP.
Quick question. So what is nitrate doing right here?
So nitrate here,
Yeah. What is it
it's what we call the final electron acceptor. So as these electrons are flowing from donor to acceptor, the last man standing in line, so to speak, the last molecule will be nitrate. So nitrate will grab those electrons, and when it has enough electrons, it gets converted to nitrite. And then nitrite gets excreted, and then hopefully there's some more nitrate in the cell, and they'll just keep this going over and over and over. Nitrate will take the electrons, get converted to nitrite, that gets excreted.
So the hydrogens are coming from that.
The hydrogens here are coming from, so when NADH gets used in this process, electrons will get stripped off of NADH and pass through the chain. That will leave you some protons to get pumped across.
Okay.
And then same here with the electron flow here, this quinine, I won't get into this, we talk about this in plant phys a lot actually, but the quinone pool here is a really nice part of the chain process where it allows for nice pumping of protons across the membrane. And so you
Is
get—
this the same as a source sink?
Not really, it's more of, so, you know, when you look at this pathway and you're like, well, why would they do this? I mean, why make it so complicated? There's all these players in the game. Why don't you just have like a one-step deal? And thermodynamically, you can't do that. Like photosynthesis too, when we cover that in my plant classes, it's this really long electron transport chain process. And it's like, why don't they just make it simpler? And it's because the cells would literally like burst into flames because there'd be so much heat release, so much energy release in a one-step process. So they break it down into these step-by-step little processes where they have this transfer of electron to this donor, to this, you know, acceptor. Then this one will give it to the quinone pool, and this one gives it to cytochrome B. And so this step-by-step and little steps process of electron transfer creates these little bursts of free energy to allow for proton pumping. So it's a very controlled way to get you some potential energy, which when I say potential energy, I'm talking about a proton gradient. So if you can get somehow strategize where you can get protons getting pumped across the membrane and get a nice, lovely proton gradient, that's potential energy or free energy that can be used. And with the microbes, a lot of what they use that for is to make ATP. Other questions on that idea?
Yeah, so like cytochrome B is right before the ATPase, if I can remember back, right? And like the process of the electron transport chain, that's like one of the carriers, right?
Cytochrome B, yes,
Yeah.
cytochrome B oxidase.
And that's carrying it to nitrate reductase now.
So this cytochrome B in this particular microbe, yep. So cytochrome B grabs the electron from the quinone pool and passes it off to nitrate reductase. And then that enzyme will take that electron and reduce NO3 minus.
Oh, I see. So instead of producing, oh, okay.
So basically the electron from cytochrome B gets given to nitrate reductase. Nitrate reductase will then allow nitrate, NO3 minus, to come into its active site, give the electron to nitrate, and that makes nitrite. And so it's this passing of electrons all the way down, in this case, to nitrate, which is the final electron acceptor. Okay, so the artist here drew on the left, if you add up how many protons, there's 5 protons being shown here and only 4 on the right. So what the artist is trying to imply here is that you get a better proton gradient if oxygen is sitting at the end. And that's true, that's accurate. You get a better proton gradient, a stronger, steeper gradient if oxygen's at the end. Yes, I see what you mean, yeah. So, and I'm not gonna get, in Gen Micro I get into it more because it's more appropriate in that class to get into a little bit more detail on this, but if you just believe me, oxygen is like the best electron acceptor. So if you have the choice, if you're E. coli and you have the choice of oxygen, you should make use of it because you'll get more ATP. However, E. coli can do what's on the right here too. You stuff E. coli in an anaerobic environment and E. coli just kind of shrugs its shoulders and says, that's okay, I'll use nitrate instead as my terminal electron acceptor. So E. coli is incredibly versatile in that regard. Yes, and so there can be, I mean, there can be some lateral movement of some of these proteins, enzymes in the membrane, but in general, you know, that one picture we saw where there's all that stuff packed into the cell membrane. So you can have like multiple systems of these embedded to allow for easy flow of electrons. So here is taking what we just saw on the right of that picture. So here is nitrate reductase. So what we were just looking at is like the front part of this. And then I just want to show that depending on the microbe, they can do more beyond that. So yes, I just showed you that you could have nitrate reductase with nitrate being the terminal electron acceptor making nitrite. But some microbes can then have these additional proteins and enzymes and in their electron transport chain system and keep it going and going and going. Where here you have the case of N2O, which is nitrous oxide. All right, so here they're taking nitrate, NO3 minus. The electron gets given to nitrate reductase, NO3 minus gets converted to NO2 minus, nitrite. And then you have this other cytochrome CD, nitrate reductase. All right, you just keep passing the electrons through these players until finally the electron gets given to nitrous oxide. Isn't that laughing gas? I think that's laughing gas. And then you get the reaction here, electron gets given to nitrous oxide and you get dinitrogen. So some microbes have this pretty extensive electron transport chain process where you can take and get, you know, instead of just stopping at nitrite, for example, they can push it all the way to dinitrogen. This is really important in some areas, some ecosystems, because this is a way to recycle nitrogen. All right, so there are some very, very special microbes that can take dinitrogen from the atmosphere and convert it to ammonia. And they hang out with plants and do what we call nitrogen fixation. And that's great for plants. However, eventually we need to get dinitrogen recycled back up into the atmosphere. So the microbes that can do this in the soil you know, depending on the soil system, this is okay because now they're able to run this reaction and get dinitrogen, which goes back up into the atmosphere. So it's I'm showing you like one half of, you know, how do we get more N2 into the atmosphere? This would be one way to go. But then also appreciate there's some microbes that will take that dinitrogen and use it and convert it to ammonia. We'll talk a little bit about that later. So what I just showed you is being shown here again, kind of simplified and showing like E. coli can do the first step, but that's as far as they can take it. So they're limited. However, Pseudomonas stutzeri, for example, can run it all the way to dinitrogen. And some of these are gases, all right, as you keep working forward. Like I said, nitrous oxide is laughing gas, and dinitrogen, of course, is a gas out in our atmosphere. So it's a way of converting, you know, all the way to the gaseous state, these nitrogen compounds. So very, very important that microbes are doing this. We need to have recycling of different nutrients, including nitrogen, in our world so that we can keep things, you know, going along. Now, if I'm a farmer, I would not want a lot of this happening in my agricultural field. It's fine if it happens in the forests per se, or in the prairie grasses, but I wouldn't really want a lot of this happening in my field if I'm growing plants. Does anyone know why?
It changes the pH of the soil.
Not so much changes the pH as when when I teach plant phys, we're actually going to get into it probably next week, mineral nutrition in the soil. So plants love nitrate. They just love it. That's their favorite form of nitrogen, most plants, most agricultural plants. And so if there's plenty of nitrate in the soil, they'll take that up and grow really well. If you have a bunch of microbes in the soil with your plants and they're doing this, they're scarfing up the nitrate instead of the plants. And so the plants will get like less nitrate. And so that can be a disadvantage. And then, Jay-Z, what were you saying? For cleanup? Yeah, so Jay-Z said, couldn't we use this if there's too much? So like if someone puts too much nitrogen fertilizer into the soil and you get an overload, and then it can run off into lakes and streams and cause pollution. So this might be a way to try to deal with that, you know, scarf up some of the extra nitrate, certainly. And that, you know, would be an easy way because microbes are pretty cheap to work with and pretty easy to get into the system. So we'll talk actually more about that too. Okay, so another concept in terms of just the basics of physiology is this one that is huge in the microbial world, but we humans, it's a very foreign concept to us because we certainly cannot do this. And so most people never even really think about this. But when you dip down into the microbial world, we know that they are fantastic at their different physiological capabilities. So here, some microbes are using inorganic compounds as energy sources. We can't do that. We have to eat something that was once living, some organic thing, whether it be a cow or some lettuce leaves. We have to take something that was organic based, that was once living, and use that as our energy source. We can't use the sun either, so that's not our energy source. So when we say energy source, we're thinking about both an electron donor, and when I say, we'll get into this idea of energy source being the idea that you run these inorganic compounds and you'll get some ATP from that, okay? So what can we think about in terms of what are the microbes doing? How do they do this? Where are they getting the inorganic compounds from? There's all kinds of sources in the environment. So anytime a volcanic eruption occurs, even just a mild belch of a volcanic eruption, it sends out a lot of chemicals that the microbes can actually use. So hydrogen sulfide, which is that rotten egg smell, that can be used as an electron source, an energy source. There's plenty of different nitrogen and iron containing compounds that, again, can be used as an electron source. And we've polluted the environment pretty nicely with a lot of those. So the microbes are pretty happy taking all that and using it. Burning of fossil fuels, anytime we do that, we create a lot of nitrogen and iron containing compounds. So again, plenty of food for the microbe, lots of industrial wastes products, biological production of just organisms doing their thing. And so basically what I'm trying to say is it really does depend on what ecosystem you're looking at, but there pretty much any ecosystem you think about, there's chemolithotrophy happening. And when you think about that, you're like, okay, inorganic, that means, inorganic means no carbon in that molecule, right? So it's like, well, they're a carbon-based organism, they still need carbon, so how are they gonna do that? Most of them will actually take in CO2 and build their carbon-based molecules off of that. You know, they'll turn it into sugar and then they can turn it into more complex molecules depending on their needs to build maybe some part of their cell. And so they're really interesting because they can take in CO2, kind of, you know, like how plants can do that through photosynthesis. Then there's this wacky group that they can't really take in CO2. They'll use a dead organism around them that's been broken down. And so that, you know, so we're a heterotroph. We eat, you know, like I said, dead cattle or, you know, dead or, you know, harvested lettuce leaves, and use that as a carbon base to build carbon in our bodies, carbon molecules. And so this last bullet point, this is a really wacky group because they don't take in CO2. So these heterotrophs, they can still grab inorganic chemicals and strip electrons off of them, but then they have to take in some organic molecule for their carbon source. So they're funky. We call them mixotrophs because they're kind of mixed up. They should, you know, most microbes take in CO2, but there's this subgroup that can't quite do that. So they're kind of, they got one foot in each world. They take some kind of organic molecule and break it down for their carbon source, but yet they use inorganic compounds as their electron donor. So they're kind of wacky. All right, so let's look at this a little bit. So here you see a lot of different reactions that have been studied. We'll actually talk about some of these as we work through microbial habitats and think about what's going on through the semester. The top one, the top line, hydrogenotrophy, I have a hard time saying that, is a very, very common strategy. What does that mean? It means they're stripping electrons off of hydrogen gas. So H2, so they take H2 and strip electrons off of it. All right, so when you take electrons off of hydrogen, you're left with protons and electrons, right? Because one hydrogen has one proton, one electron. So if you have two hydrogens, then it's two protons, two electrons, all right? So what they're implying here, this is a very shorthand way of writing this, but this last column, what they're implying here is that there is some kind of electron transport chain process going on. In this case, oxygen is the final electron acceptor. And so as hydrogen is getting stripped of electrons, eventually those electrons end up at oxygen, and eventually water will get produced. All right, so you can see if you look on the right column, oxygen is a common strategy to have oxygen as the terminal electron acceptor, but not always. All right, but again, think of it like a game. These microbes are doing this because of the selection pressure of their environment. They were put into an environment where there's plenty of hydrogen gas. And so they evolved the mechanism to strip electrons off of that, play the game of running a short electron transport chain because that gets me my proton gradient, which gets me ATP. Right? They don't really care about oxygen per se getting converted to water. That's not the goal. That's not really what they care about. They're just playing the game so that they can create this potential energy of, I want to get a proton gradient so that I can do something with that potential energy. And very often it's to make ATP. So lots and lots of different options here. Hydrogen gas is common. You see iron, some nitrogen containing molecules. Methylotrophy is a really interesting one too. And then iron oxidation, we talked a little bit about that actually, as you know, iron was super abundant in the early Earth. And so that was probably a very primitive first reaction. So hydrogenotrophy is super common. And so that's a really good example because hydrogen gas is pretty darn good at donating off electrons. And so it has what we say good reducing potential. It's very, very, chemically speaking, I like to think of it as it's very happy to donate off electrons. It's a very enthusiastic electron donor, all right? And so the end result of that is you can get some pretty good ATP production through this process. So hydrogenotrophy, if a microbe can do that, and if they're presented with hydrogen gas, that would be a good strategy. It'll get them some nice ATP. So let's just look at this, like how do they actually do it? So I put this here not to have you to memorize step by step, but to think about, okay, what are they doing here? So here's your hydrogen gas here. So this microbe has the necessary hydrogenase enzyme to strip electrons off of hydrogen gas. And you can see it's an electron transport process. You have all these players donating and accepting on down the line. And in this process, you get proton pumping occurring. Okay, you get this proton pumping, you get free release of protons as you strip electrons off of hydrogen. So this should look vaguely familiar. We looked at this, at least this part, way back in week 1, about this idea that if you strip electrons off of hydrogen, you can feed it in. In this case, it's going into an electron transport chain, get this nice proton gradient established, protons diffuse back through the ATPase, and you get your ATP. Here you see CO2, so this particular microbe, even though it's stripping electrons off of this inorganic hydrogen gas, it's able to take in CO2 and run reactions where they just say cell material. So that could be, usually it turns into a sugar first, like glucose or something like that, and then it can take that and run with it and make other necessary products it needs to build its cell, repair any damage, whatever. Okay, so chemolithotrophy. Lithotroph, if you are a lithotroph, litho means, if you look that up, it means rock, like, you know, rocks. Troph means to eat. And so if you're a chemolithotroph, you eat rocks, which means you're capable of taking and stripping off electrons off of some inorganic molecule. Okay, why didn't I want to go up there and have it. Wow. All right, I saw a, come on. Why won't it allow me to stop share? Why can't I stop the share? I was trying to find the chat box. Okay. Let me look at the chat real quick. Someone chatted in. Yes, I did. I did. Thank you. I took attendance earlier. So we got to stop there, I guess. We're out of time. But I just want to touch on photosynthesis. Again, I'm going to hit the highlights of photosynthesis. I'm certainly not going to go into any depth as what I do in plant phys. But we have to appreciate that microbes can do photosynthesis similar to plants. But then there's a whole other group that do this wacky kind of photosynthesis, very different from plants. And so I need you to think about both things happening. But we'll do that next time. I guess tomorrow, right? We'll be all Zooming tomorrow. Okay, we all Zoom in tomorrow. I got some, let me stop recording.